Thank you to the organizing committee for selecting this abstract for presentation and for persevering in terms of hosting this virtual conference despite the challenges that it must bring. So unfortunately, unlike Philip's really amazing work, I don't have mechanism. We really have preliminary results from humans, but hopefully what I want to show you is perhaps some of the methodology that you may consider when trying to assign relevance and maybe prioritize which bugs may be of interest in your disease cohort. Next slide, please. Great. Okay, so the disease that I'm working on is actually called systemic sclerosis. And these patients are quite interesting to us because they're autoimmune in nature. The disease primarily affects connective tissue, the skin, and the organs. And this is important because this is the main, I guess, damaging feature is that fibrosis in these patients, because of chronic inflammation, leads eventually to organ failure. The etiology is unknown. However, in some patients, immunosuppressants are effective, and this points towards the fact this may be an autoimmune disease. In addition to immunosuppressions being effective, we also see the expansion or appearance of autoantibodies that are specific for these types of patients. And what I want to highlight here is that there's a high patient-to-patient variability. So this is quite common in complex inflammatory diseases. And what's interesting to us is that the gastrointestinal complications are common in 90% of the cases. So this really pointed to the question, how does the gut influence perhaps the systemic inflammation we see in these patients? Next slide, please. And what I'm showing here is a study that we published last year. It was really led by the three individuals on the left, Oven, Anna Maria, and Hovard, who completed his PhD earlier this year. And it was a fecal microbiota transplant in advanced SSC patients. And this is the design of the study and really formed the rationale for the work that I will show. So we had 10 patients originally, this small pilot, and 5 had FMT. And this FMT material was a transfer of anaerobic cultivated human intestinal microbiome, also known as ACHIM. So many of you are familiar with this. It's basically a standardized, commercially available microbiome that people have used in various settings to reduce inflammatory conditions. And so we had 5 of these patients and we treated them at week 0, week 4, so twice, and then we followed them until week 16. And conversely, we had 5 patients initially that only, in the end, had 4 that received placebo controls. And what we saw, please, next slide. Sorry, just waiting for the slide to advance. Great, thank you. What we saw is we focused primarily on the lower GI symptoms, as 90% of the patients have reported GI symptoms. And at week 0, what I'm showing here is the baseline state at which they experienced such symptoms. So we had 8 categories that are clinically relevant. I'm focusing only on 2 here, bloating and diarrhea. And we saw in the majority of patients, both in placebo and control at baseline, had similar levels of lower GI symptoms. Next slide, please. Oh, sorry. That's fine there. And what we're showing here is at week 4 and week 16 that we saw improvement following FMT. So week 4 is 4 weeks after the initial FMT, and week 16, this is, I guess, 12 weeks after the last FMT. And you can see that there's a quite substantial improvement in these lower GI symptoms. So what we concluded is from the small pilot that FMT with a standardized mixture of this ACHIM mixture bacteria reduced lower GI symptoms in these patients. Next slide, please. And during the course of this experiment, we also looked, of course, at the microbiota composition, and we showed that there are some changes, particularly in these Rheumococca and Lanctospira, But really, we don't know what the significance of these are because it's so difficult with the 16S sequencing to really assign mechanism and causality to these one time point studies. But this kind of justified to us that maybe we should look more into the lower GI and how the microbiome affects inflammation in these patients. Next slide, please. And so this really formed the background. So we saw that FMT reduced lower GI symptoms in advanced patients, and this supported our hypothesis that there's a role for the gut microbiome in disease. But the question really that arose from this is gut dysbiosis seen prior to immunosuppressive treatment in these patients? So again, I mentioned these are advanced patients that receive this FMT. What if we look on early? And this really formed the hypothesis that early SSC patients will also have an altered gut composition. And we define early as average 13 months from onset of symptoms. And we are able to collect patients from an Oslo cohort, a Lund cohort, and healthy controls. And then we ran these fecal samples through 16S RNA sequencing and looked at relative abundance. Next slide, please. And what I'm showing here is a phylogenetic tree of the bacteria that we detected. And how we're looking at this, or how this can be interpreted, is that on the baseline in the gray is the Oslo cohort, and overlaid on top of that is the Lund cohort. And the first question we want to ask is there any differences really between these two independent SSC cohorts? And we did see some increases and decreases. We saw an increase in Provotella and a decrease in this Corynebacteria. But for the most part, our conclusion from this analysis is that the early SSC microbiome is similar between two independent cohorts. Next slide, please. And so our second aim really was now to focus on the Oslo cohort versus healthy controls and the Lund cohort versus healthy controls. And in the Oslo cohort, surprisingly, we did not see any differences between early SSC and healthy controls, whereas in the Lund cohort, we did see some differences. So these are a little bit contradicting, but what this may suggest is that dysbiosis in advanced disease is possibly a secondary effect and not disease causing. Another possible interpretation of this data is that small changes in rare subgroups are difficult to see in a global analysis. And this is actually probably nicely highlighted in the previous talk by Philip, that maybe you need to focus on particular bacteria species or you won't see changes. Next slide, please. And so this formed our second aim. So we showed, my background actually is in B-cell immunology. And so we used what was known as bug facts, as Marius alluded to, and which presented by Dr. Desai in the keynote presentation, where the group PALM showed that if you look at IgA-coded bacteria and transfer it to disease-susceptible models, you have worse disease. And so that, I guess, flavored our experiments, and we were looking at these immunoglobulin or Ig-coded bacteria and asked the question, do we see any differences in early SSC? Having the hypothesis that, yes, we would detect differences because this is an inflammatory disease. Next slide, please. And so what we had here is, again, our early cohorts from Oslo and Lund and healthy controls. And this is the basic methodology we use. We had snap frozen fecal pellets, we homogenized, then we stained with either IgA, IgM, or IgG. And then we sorted using flow cytometry and then performed 16S. So what I'm showing here in the middle is a representative flow cytometry plot showing Ig-coded bugs on the top and Ig-negative bugs on the lower quadrant. And we focused on IgA and IgM and IgG because IgA is the most secreted antibody in the gut lumen. IgM is thought to be kind of a more naive response, so the initial response, whereas IgG is highly specific, high affinity, and may denote high affinity, or sorry, potentially pathogenic members of the gut microbiome if we saw an expansion of these. Next slide, please. And so what we saw here is, again, we focus on IgA, IgG, IgM, and IgG. And for the most part, I'm just focusing on the differences that we saw across the both cohorts. And so we saw there's increases in three main bacteria, this Delsulfide Vibrio, Veillonella, and Streptococci. And what's interesting, and of course we don't have causality, we haven't put these into an animal model yet, but it's interesting that other groups have shown that these bugs are associated with lower GI inflammation and fibrosis of various tissues. And the main point, other than the bugs that I want to highlight, is that I think this underlies, again, that Ig coding perhaps can be used to highlight key bacteria that may otherwise be overlooked in an unbiased global analysis. Next slide, please. Okay, so for the final aim of the study, because we had these early SSE patients, we wanted to know, does immunosuppression actually also affect the microbiome? So we stratified we combined the Lund and Oslo groups and stratified them based on whether they had immunosuppressive treatment, which is standard of care, or they were untreated, treatment naive. And then we sorted the bugs and we looked at the composition using 16 RNA. And what I'm showing here is, again, we're just focusing on the ones that overlap, that there are similarities between the bugs that are found in IgA, IgM, and IgG. Their significance still is to be determined, but it shows that indeed, if you stratify your microbiome based on Ig coding, there's a possibility that you may be able to focus on bugs that may be more inflammatory to the immune system. Next slide, please. And so in conclusion here, to say that we didn't see any differences in the overall gut microbiome, at least in the Oslo cohort compared to the healthy controls. And again, this suggests that maybe there aren't that many changes early on, and that the changes in the gut microbiome that we see in these patients are secondary, and they may actually amplify existing inflammation. The second conclusion is that the sorted Ig-coded bacteria allows us to see greater changes in the global microbiota. And we see these changes, these bacteria associate with an increased abundance of clinical features that seem to be relevant in this disease cohort. And lastly, unsurprisingly, but something we wanted to confirm, is that immunosuppressions do alter the composition of the gut bacteria. And this is just to highlight that that we should keep in mind that the treatment and when we collect the samples in which part of the disease history is very, very important and can affect the gut microbiome. Lastly here, next slide, please. I wanted to highlight two limitations in future, or one limitation, one future direction. So I mentioned that we use this Ig coding as perhaps a better way of finding important bugs. And what I'm showing on the left here, this flow cytometry plot, one thing that we encountered again and again was, how do we determine what is actually coded? You can see here in the previous data, I had a simplified image where we had positive bugs and then we had negative bugs. But really each sample is quite different. In some samples we have this discrete groups, but in many samples we have this smearing or this spectrum of coding. And so we're exploring now whether there is any difference between bugs that are highly coded versus intermediate coded and negative. And then secondly, I wanted to say that one of our future directions is determining mechanism and causality. And one of those is perhaps looking at this immunocoding index. And this is mentioned also by Dr. Desai in his keynote presentation, this ICI. And really what we're trying to look at is, if we look at the figure down below, we have pre-treated versus post-treated, where the negative fraction changes after an intervention. And perhaps the change is actually more important than whether it is coded initially. And so we're trying to use machine-based learning. This is really a collaboration that we're leveraging with Victor Greif, who's a group leader here at Oslo University, to see whether there is any metric that can better stratify what are important bugs and what aren't. On my last slide here is my acknowledgements. I really like to thank all the people, particularly my supervisors, Johannes and Tom Carlson, and also, of course, Anna Maria, Oven, Håvard, and Henrietta, who did staining for the study, and Victor, who's helped try and refine this ICI index. And thank you very much for your attention.
Thanks a lot. That was very, very interesting, I think. And it's also interesting to see that when you do normal 16S, you do not get so much information, but when you combine it with IG staining, you really are able to find the sulfovibrio, which we previously found to be HIV related, and you find Veillonella, which Johannes' group found to be PSC related, and it's really possible that these bugs really mean something. But the question is, of course, as you also said, that this scatter plot, it's a big cloud. So where do you set the cutoff? And is it possible to refine this to, for example, use multi-channel flow cytometry and stain for combinations of immunoglobulins and so on to be a bit more sure that these are really Ig stained?
Yeah, that's a great question, Marius. I would say actually, no, it's probably impossible using patient samples and asking a question directly, staining the patient samples and seeing what appears. I think probably the reverse strategy may be more useful in determining, or I guess excluding non-specific bugs. And the reverse situation, I mean, is that we specify which B cells we think are important and find the antibodies then, or generate the synthetic antibodies, and then look at feces that is uncoated. So we start from a platform that is unlittered or clean from host immunoglobulin, and then we try and identify, okay, in these patients we know there's antibody produced, and then we can try and look at which bugs these antibodies recognize. So I think it will be always very messy if we try and use patient samples. But I think if we use the reverse, so we use maybe a synthetic microbiome, as was highlighted today, so that's of course a model, but at least that's something we can define. And then at least in our situation where we're trying to look at antibody coding, we can really exclude non-specific binding.
Thank you. Johannes, any questions from the live feed?
Yes, I have a question. It was regarding the FMT study. Did you explore if the bacteria in the ACHEM, the bacterial mix that you gave to the patients, if it actually settled in the gut?
Yeah, so that's also a great question. So the question is, do we see any of the bacteria there start to colonize the gut of our patients? And we did see some evidence of this, but because majority of the bacteria that we detect in ACHEM is found also in patients, it's hard to then again discriminate, is this change really due to colonization of ACHEM, or is it just simply a change of the existing bacteria? So there weren't any bugs that were specifically increased or decreased that only originate in ACHEM. And this is also something that we're exploring now.